Hey, welcome! We hear so much about the gut microbiome, right? This bustling city of bacteria living inside us. But what if I told you we've basically just been looking at a city map without knowing anything about the people actually living there? Well, today we're diving deep. We're going to use some incredible data to pull back the curtain on a hidden layer of our inner world. Let's get into it.
Okay, so our first big idea. It's a subtle one, but it is absolutely crucial. For years, scientists have talked about the microbiome in terms of bacterial species. But to really truly understand what's going on, we have to get way more specific than that. So take a look at this. This slide really sets up the mystery for us. Scientists were studying this super common gut bacteria called Prevotella copri, and they noticed something weird. You could have people on completely different diets, omnivores over here, vegetarians and vegans over there, and they all had this same species of bacteria. But look how their microbial communities behaved in totally distinct ways. You can see how they're clustering into separate groups. And that leads to the big question, right? If everyone has the same bacteria with the same name, what on earth is driving these massive differences? The differences that so clearly separate people based on something as simple as what they eat. And here it is, the big reveal. It's not about the species, it's about the strain. Think of it like this, okay? You and your neighbor might both have a dog. The species is Canis familiaris for both of you. But you might have a tiny little Chihuahua, and they've got a giant Great Dane. See? Same species, but totally different versions or strains. That's the difference we're talking about. Right, so if strains are the secret sauce, how in the world do scientists tell these microscopic versions apart? I mean, we're talking about a sample with trillions of bacteria. To figure that out, we've got to look at the incredible tools they've built. The technique is called strain profiling. And really, it's just a fancy name for a set of super powerful data science methods that act like a genetic magnifying glass. They let researchers zoom right past the species label and pinpoint the tiny, specific genetic quirks that make each bacterial strain unique. So one of these methods is called PanFlan, and it's basically genetic detective work. It's so cool. It starts with this jumbled mess of DNA from a gut sample. That's what you see on the left. And it maps all that data against this huge library of known bacterial genomes. By figuring out which genes are there and which ones are missing, the tool can identify the unique genetic fingerprint of the exact strain that's in your sample. This is a huge leap forward, no question. But so what? What does having this incredibly high-res view of our gut actually let us do? Well, let's open up a few real-world case files and see just how big the impact is.
First up, this one's serious. Public health. During a really dangerous E. coli outbreak in Germany, this kind of strain-level analysis was a total game-changer. It let scientists zero in on the exact genetic features, things like toxin production or antibiotic resistance, that made this one particular E. coli strain so nasty and separate it from the countless harmless strains living in our gut every single day.
Next up, making medicine work better. For a procedure like a fecal microbiome transplant, or FMT, the whole thing works only if the good donor bacteria actually survive and set up shop in the patient's gut. Well, strain profiling can now help predict which donor strains are most likely to successfully engraft to make a new home, making this powerful therapy way more reliable. And get this, it even helps us discover brand new biology. A study in Bangladeshi children found this unique strain of Bifidobacterium longum. And what made it so special? Well, it had this super rare talent to digest sugars from both human milk and plant fibers. It's like a transitional bug perfectly designed for a growing kid. This could literally be the start of a whole new generation of probiotics. So, okay, we can now identify the individual players with amazing precision. But that's really only half the story, isn't it? Because in the microbiome, no bacterium is an island. They don't act alone. So what about their connections, their relationships? And that brings us to the next mind-blowing level of complexity, the microbial social network. Our gut isn't just a random bag of microbes. No, it's a complex, deeply interconnected community. It's a network where the interactions are everything. And the health of this network? It's absolutely critical to our health. When these connections get messed up, a state that scientists call dysbiosis, it's linked to a whole laundry list of chronic diseases, from Crohn's disease to obesity, even some cancers. So understanding the network is really about understanding health and disease. This slide shows it perfectly. Microbial networks aren't random, like that tangled web on the left. Instead, they're what's called scale-free, like the network on the right. And what that means is that they have a few super connected hubs. See those big red dots that are way more important than everybody else. These are the key influencers, the linchpins holding the entire community together.
So the goal becomes pretty clear, right? How do we map this invisible social network? And maybe more importantly, how do we find those key players? How do we identify those super important hubs?
Well, to do that, scientists use another brilliant data tool. This one's called WGCNA, which stands for Weighted Correlation Network Analysis. Its entire purpose is to sift through mountains of data to find the cliques, the little groups or modules of microbes that always seem to act together.
And the output looks something like this wild, colorful grid. It's called a topological overlap matrix. But don't worry about the name. The only thing you need to see are those bright squares running along the diagonal. Each one of those squares represents a clique, a tight-knit community of microbes that are so interconnected they basically act like a single team.
And this method is so powerful, it can take the behavior of an entire team, one of those bright squares, and summarize it into a single signature. They call it a module EGON gene. This is a huge deal. It lets us go from trying to track thousands of individual bacteria to just watching a handful of functional teams, which makes understanding the whole ecosystem so much easier.
All right, let's bring it all home. We've gone on this incredible journey, from species down to strains, and then from single microbes all the way up to their entire social networks. So what does this all mean for you, for the future of medicine?
What it really means is that we've developed the tools to go from looking at a sea of jumbled DNA to creating a crystal clear picture. A picture where we can point to individual actors, understand exactly what they can do, and then map their entire web of relationships.
And the future this opens up is just astounding. We're talking about personalized medicine based not just on your human genes, but on your unique microbial strain profile. We're looking at predictive tests that can spot the earliest signs of disease just by seeing a shift in your microbial network. And it means we can finally develop really smart, targeted probiotics and therapies that support the health of our entire inner ecosystem.
And that leaves us with one last, really powerful thought to chew on. What if we could predict your risk of developing a chronic disease years, maybe even decades in advance, just by mapping your inner social network? That is the incredible future that these data science tools are helping us build, one microbe at a time.